All right, so the exam. So remember, this one is next Thursday at 6 o'clock. It's going to take us a bit over 2 hours. So I'm budgeting like 2.5 hours. If we go a little bit over that, that's fine. No skin off my nose. Um, but it's a big exam and it covers a little bit of everything. This study guide I made literally from the exam. So it has essentially kind of the main topics are going to be that it covers, and then about how many questions are going to be on each system, things like that. So it's a nice study guide. It's going to be quite useful for you. So as I said, my plan for tonight is to just kind of go through this entire thing. It's going to take as long as it takes. If we go past 8:50, go to sleep and just watch the recording. Okay. Let's see, and once again, if you have any questions at any point about anything, we will kind of work on that together as well. But I'm essentially kind of reteaching the entire course tonight. It's kind of what comes down to, exactly what you kind of need to know for the final. So hopefully that you'll find that useful. All right, so let's start at the beginning. Oh, and the exam is going to go in this order too. So it's not going to be all like jumping around all over the place. It's literally in the order of that we cover the topics. So kind of study it in this order, go over it in this order, and you're going to see it in this order on the exam as well. So it's not going to be kind of randomized. It's literally going to be going through these topics one by one, is the good news. All right, so let's talk autonomic nervous system, because that's literally the very first thing we did way back at the beginning of the course. So, all right, so remember we have kind of multiple nervous systems or multiple branches to our nervous system. Our two big ones we talked about were the somatic and the autonomic nervous system. So know the differences. So remember, the somatic nervous system is voluntary, conscious control of our skeletal muscles, right? So that's essentially how we tell our skeletal muscles what to do. The skeletal muscles are the effectors of the somatic nervous system. The way our central nervous system communicates with our skeletal muscles is through a single highly myelinated neuron that makes it nice and fast, right? So once again, somatic nervous system, voluntary conscious control of skeletal muscles. It has one neuron going from our central nervous system to the skeletal muscle, which is the effector. And that one neuron is highly myelinated. And that one neuron, does anybody remember what neurotransmitter is released by our motor neurons to our skeletal muscles? ACH, exactly, acetylcholine. Thank you, Karina. Wonderful. All right, so that's our somatic nervous system. So our autonomic nervous system is going to be different, right? So instead of being conscious control of skeletal muscles, autonomic nervous system, remember, automatic. It is unconscious control of all of the things that affect homeostasis. So like digestion, heart rate, respiratory rate, things like that. So with our autonomic nervous system, once again, it's unconscious. Instead of just one highly myelinated neuron going to the effectors, in the autonomic nervous system, remember, it's two neurons in a chain. We have a preganglionic neuron and a postganglionic neuron, right? The preganglionic neuron is lightly myelinated. The postganglionic neuron is going to be heavily myelinated. Sorry, unmyelinated. Stop. So once again, preganglionic neuron, lightly myelinated, postganglionic neuron, unmyelinated. Problem with taking cold medicine, gang. It makes me a little loopy. I apologize. All right. So the effectors are also going to be different in the autonomic nervous system, where it's skeletal muscle in the somatic nervous system. The effectors in the autonomic nervous system are cardiac muscle, smooth muscle, and glands. So once again, autonomic nervous system effectors, cardiac muscle, smooth muscle, and glands. So let's go over the features once again. So once again, autonomic nervous system, unconscious control of essentially homeostasis, right? Regulates digestion, regulates heart rate, regulates respiratory rate, all that good stuff. Instead of one neuron, we have two neurons in a chain. Preganglionic neuron, which is lightly myelinated, a postganglionic neuron that is unmyelinated. And once again, the effectors are different as well. Instead of skeletal muscle, it's cardiac muscle, smooth muscle, and glands. All right. Functions of the autonomic nervous system. So once again, homeostasis, maintaining homeostasis, making sure the different parts of the body get what they need during different situations. And that kind of brings us to our sympathetic and parasympathetic nervous systems. Because remember, the autonomic nervous system itself is divided into two subsystems. So the sympathetic nervous system, remember, that's fight or flight. So it gets us ready for emergency, gets us ready for excitement, gets us ready for exercise. Okay. So essentially, we're going to be making sure we provide resources to our skeletal muscles so they can do their jobs. We're going to make sure that our lungs are working at their most efficiently. We're going to make sure our heart is working at its most efficient and most rapid status. So effects of sympathetic nervous system activation specifically, as we kind of move down our, kind of cross these off as we go. Let me just go to the highlighter. Good. So once again, effects of sympathetic nervous system stimulation or activation. Well, you tell me. So if our sympathetic nervous system is activated, what's going to happen? Heart rate's going to increase. Respiratory rate's going to increase. We're going to see epinephrine and norepinephrine released into the bloodstream from our adrenal medulla. We're going to see our blood sugar levels increase. Things like that. Okay. These are all things that are going to kind of increase our ability to maintain physical activity. Things like that. Parasympathetic nervous system, on the other hand, is going to have the opposite effects. So remember, parasympathetic nervous system, its activation is, remember, rest and digest, right? So what's that going to do? Well, that's going to increase digestion or digestive absorption, whereas sympathetic would decrease it. It's going to decrease heart rate. It's going to decrease respiratory rate. We may actually feel kind of tired and sluggish and lethargic during parasympathetic activation, right? So these are kind of important effects. Remember that they also release different hormones, right? Or different neurotransmitters, I should say. So sympathetic nervous system, at its effectors, it's going to release norepinephrine. The parasympathetic nervous system, at its effectors, is going to release acetylcholine. Remember that the sympathetic nervous system activation is going to be much longer lasting and more diffuse, because besides just releasing norepinephrine at our effectors, it's getting dumped into our body by the adrenal medulla. So remember, not only are we releasing norepinephrine during sympathetic activation directly to the effectors through the neurons, it's also getting flooded through our system by the adrenal medulla as it releases it into our blood, and it goes all over the place. So once again, good question, hint, hint. That likes to be asked is, okay, why is sympathetic nervous system activation so long lasting? Well, it's because norepinephrine gets released as part of it, and it doesn't break down very quickly, unlike acetylcholine with sympathetic activation. All right. Let me think. So remember, both sympathetic and parasympathetic, once again, are autonomic parts of the autonomic nervous system. So once again, we always have two neurons going to their effectors, but we also have ganglia in between our preganglionic and postganglionic neurons, right? So the sympathetic nervous system has ganglia that are closer to the spinal cord. So our sympathetic nervous system has prevertebral and paravertebral ganglia, which are close to the spinal cord. The parasympathetic nervous system has terminal ganglia, which are located on or near the effector organs. So those are the three main ganglia you need to know about. So remember, sympathetic, prevertebral, and paravertebral, which are very close to the spinal cord. Parasympathetic, the ganglia are located really very close to the effector organs. All right. Let's see what else we got here. Preganglionic and postganglionic neurons. So preganglionic neurons, remember, lightly myelinated. Preganglionic neurons always release acetylcholine. The effect of that acetylcholine is always going to be stimulatory. Postganglionic neurons, remember, are going to vary depending on if they're sympathetic or parasympathetic. Sympathetic postganglionic neurons are always going to release norepinephrine to the effector organs. Remember, the effects of norepinephrine are always can vary. It depends on the organ. It depends on the receptor. It could be stimulatory. It could be inhibitory. What do I mean by that? Well, for example, if we release norepinephrine to our heart, that's going to have a stimulatory effect. Our heart rate's going to increase. If we release norepinephrine from our parasympathetic nervous system at our digestive system, that's going to decrease digestive system activity. So once again, depends on the organ, depends on the receptor. Same thing with our parasympathetic nervous system. Sometimes it's going to release at its postganglionic neuron acetylcholine, but once again, the effects are going to be either stimulatory or inhibitory depending on the effectors. Let's see. We've kind of covered ganglia and their locations. So we'll cross that out. We've talked about preganglionic and postganglionic neurons and their similarities and differences. We've been talking about neurotransmitters. So the location of the secretion and the effects. So once again, our two main neurotransmitters when we're talking about autonomic nervous system are acetylcholine and norepinephrine. Acetylcholine gets released by our preganglionic neurons, all of them. The effects are always stimulatory. In the sympathetic nervous system, norepinephrine gets released from our postganglionic neurons. Its effects are stimulatory or inhibitory, depending on the organ and the receptors. Our parasympathetic nervous system releases acetylcholine at the effectors. And once again, the effects can be stimulatory or inhibitory, depending on the receptors, depending on the organ. Things like that. Let's see. Anything else I can think of with that stuff? We talked about the effectors, we talked about the functions. Visceral reflexes. Let's talk about that quick, and then I'll collect my thoughts and kind of think about those. So visceral reflexes are, remember, how the autonomic nervous system works to maintain homeostasis. So essentially, something senses a change within our body, and the autonomic nervous system responds through these visceral reflexes. So good examples of this, like our baroreceptor reflex, where if our blood pressure gets too low, pressure receptors in our carotid arteries and places like that will sense that, send a signal to our hypothalamus, which will send a signal through our sympathetic nervous system to essentially increase cardiac output, increase blood vessel constriction to raise blood pressure. Another good visceral reflex would be like in digestion. So when food enters your stomach, a visceral reflex will increase the production of things like gastric juices, stomach acid, and enzymes and things like that. So once again, it's got to be some sort of neural control of actual cardiac muscle, smooth muscle, or glands, right? It's not the same as ducking out of the way when a ball flies toward your face or your patellar-Diedrich reflexes. Those are somatic reflexes. Visceral reflex that affects skeletal muscles. These things, visceral reflexes always affect cardiac muscle, smooth muscle, or glands. So make sense to everybody?
Yeah.
Okay, any questions so far, Greg? I know this is a little bit of a blast from the past. One other thing to remember, as I'm thinking of it. So when we talked about sympathetic versus parasympathetic nervous system, remember sympathetic nervous system, we refer to it as thoracolumbar because the nerves of the sympathetic nervous system come out of the thoracic and lumbar vertebrae. Our parasympathetic nervous system is considered to be craniosacral because the nerves come out of it's either the cranial nerves or the sacral nerves for the parasympathetic system. So remember, parasympathetic system, craniosacral, sympathetic nervous system is thoracolumbar. All right, I think that's pretty good. Any questions on autonomic nervous system while I just really quickly blow my nose? Hope I got the mute button on in time for my sneeze. All right, so endocrine system. So do you remember the endocrine hormone sheet we had from the beginning of the semester that had all the endocrine hormones and their kind of functions and what they targeted and things like that? Make sure you look that over. Okay, because there's going to be a goodly amount of hormones and their targets and their effects in there. So keep that in mind. Once again, overall functions of the endocrine system: maintain homeostasis, promote growth, development, things like that. It's kind of the endocrine system's overall kind of function. Remember how these things work. So remember what hormones are. Hormones are chemical messengers that are released into the blood. Once they're released into the blood, they travel throughout the body. They bind to their specific receptors and alter the activities of their targets or of their target cells, right? Remember, some hormones are very, very targeted. So like FSH and LH only affect your gonads. Thyroid stimulating hormone only affects your thyroid gland. Other hormones are very broadly acting. Our best example of a hormone that literally affects every cell in your body is thyroid hormone. Okay, thyroid hormone literally affects almost every cell in your body because every cell in your body has receptors for it. Okay, and it's our main hormone of metabolism. Control of hormone secretion. So remember, we can have neural control where actual nerves go to the glands and tell the glands to release their hormones, usually nerves coming from our hypothalamus. Good example of this would be our hypothalamus communicating with our posterior pituitary to release ADH and oxytocin. Maybe our hypothalamus communicating directly to our adrenal medulla to release epinephrine and norepinephrine during sympathetic nervous activation. We can also control hormone secretion through what we call humoral control. Levels of nutrients in the blood will affect what hormones get produced. Good example of this is like calcium homeostasis. So remember our two hormones that control calcium levels in our blood: parathyroid hormone and calcitonin. So remember, if calcium levels get too low in our blood, we're going to release parathyroid hormone. That's going to raise blood calcium levels. If calcium levels get too high in our blood, we're going to release calcitonin. That lowers blood calcium levels. So remember, those are our two main calcium affecting hormones. Steroid hormones. So remember, steroid hormones are lipid soluble hormones, right? Our steroid hormones are all derived from cholesterol. Our steroid hormones can't travel through the blood by themselves. They have to be transported by carrier proteins. But the most important thing to remember about steroid hormones is it's kind of how they act. So remember, because they're lipid soluble, they can diffuse directly into the cells and essentially trigger what we call direct gene activation. So they bind to receptors inside the cells, and those receptors activate genes to make proteins. So that's how steroid hormones work. In contrast, we have water-soluble hormones, right? Water-soluble hormones can't, right? They can't actually diffuse into the cell. So water-soluble hormones have to bind to receptors on the cell membrane and activate what we call the second messenger system, where it starts kind of a chain reaction of different proteins activating each other. And remember that our second messenger was cyclic AMP or CAMP. That was our main second messenger that we talked about. So remember, steroid hormones, direct gene activation, water-soluble hormones, like our peptide hormones or an amino acid-based hormones. Remember, they have to bind to receptors on the cell's surface and activate the second messenger system. Okay. Hormone interactions. So once again, we have synergism, where two hormones kind of multiply each other's effects. They have a multiplicative effect. They have essentially by themselves, they have generally the same sorts of effects. A good example of synergistic hormones would be things like glucagon and epinephrine, which both raise blood sugar levels. Same thing with cortisol. Permissiveness, where one hormone has to be present for another hormone to be able to actually do its job to work. A really good example of this is thyroid hormone and growth hormone. For growth hormone to be able to affect cells, thyroid hormone also has to be present. And then finally, we have antagonism, where two hormones have opposite effects. So insulin and glucagon are good examples of these. Insulin lowers blood sugar levels, glucagon raises blood sugar levels. Parathyroid hormone and calcitonin also good examples of these. Parathyroid hormone raises blood calcium levels. Calcitonin lowers blood calcium levels. So yeah, let's see what else is. What I think of. We talked about permissiveness. Talked about steroid hormones. Being lipid soluble and able to pass through membranes. Once again, remember your kind of general hormones, what they do, their functions. So like things like growth hormone you should know about, where it comes from, what it does. Thyroid hormone, where does it come from? What does it do? What stimulates thyroid hormone production, which is thyroid stimulating hormone. Be able to match like the hormones with their functions or the glands they secrete. Things like that. So there's a goodly amount of questions on that 16. So make sure you look at that. Any questions? So that kind of covers all the stuff that we've done so far. Any questions on endocrine system? Anything you want me to go over again? Either second messenger system, things like that.
What was the example of
So permissiveness, yes, absolutely. So the example of permissiveness is that growth hormone can't do its job unless thyroid hormone is present. So once again, so growth hormone can't do anything unless there's an adequate amount of thyroid hormone also circulating in your blood and down to cells. So that's why, if you remember, like when we talked about like thyroid hormone disorders, that's why like children and infants that don't have enough thyroid hormone, they get a form of dwarfism. They end up very short and small in stature. Because without the thyroid hormone, they're producing plenty of growth hormone. It just can't do its job without the thyroid hormone being there as well. Okay.
Professor, can you give the example that you gave for
Synergism, maybe?
Synergism, absolutely. So synergism is, remember, when two hormones with similar functions kind of multiply each other effects. A really good example of that would be like glucagon and epinephrine. They both
Okay.
raise blood sugar levels, and if they're both present, they're gonna raise blood sugars a lot more than either of them by themselves.
Okay, thank you.
You're very welcome. And as I said, antagonism, like the two main ones are insulin and glucagon. Insulin, once again, lowers blood sugar levels. Glucagon raises blood sugar levels. That's our two best examples. Let's see. Any other questions on endocrine system before we move on to cardiovascular? All right, so let's talk cardiovascular system. So first thing, erythropoiesis. So remember, that's red blood cell production. Remember, the hormone that actually triggers red blood cell production is erythropoietin. So red blood cell production is erythropoiesis. What triggers it is the hormone erythropoietin, which is produced by your kidneys, of all places. So once again, so erythropoiesis, the thing I want you to remember is it's erythropoietin that triggers it. Okay, that's the hormone that triggers red blood cell production.
Can you say that again? I just missed that.
So once again, the
sorry.
hormone, yeah, no worries. That's what I'm here for. So the
And-
hormone erythropoietin, and actually let me type it on there. Let me just get it back to that's our hormone erythropoietin that does red blood cell production. All right. So heart anatomy and dysfunctions. Now the thing is, gang, this is a final, so it actually combines a little bit of what you learned in lab with what you learned in lecture. Okay, so we're gonna kind of refer to some stuff you learned in lab 2. So like heart anatomy and the functional anatomy of the heart. So for example, blood flow through the heart, what the different valves do, things like that. So let me give you an example. So let's say our tricuspid valve. So our tricuspid valve, right, is on the right, correct? So it's between our right atria and our right ventricle. What do valves do? They prevent what?
Backflow.
Backflow. Right. So for example, our tricuspid valve will prevent backflow into the right atria when the right ventricle is contracting, right? Just like the bicuspid or mitral valve will prevent backflow into the left atria when the left ventricle is contracting. Just like our pulmonary valve will prevent backflow from our pulmonary trunk into our right ventricle when the right ventricle relaxes. And our aortic valve will prevent backflow from our aorta into our left ventricle when our left ventricle relaxes, things like that. So think of like, okay, think about like where blood's going to flow. Think about what the valves are going to do. Think about like oxygenated versus deoxygenated blood in the heart. So remember, our right side of the heart typically has deoxygenated blood because it's receiving deoxygenated blood from the rest of the body through the veins. Our left side of the heart is going to be full of oxygenated blood because it just received all the oxygenated blood coming back from the lungs through our pulmonary circulation, things like that. So not a bad idea to just kind of review some heart anatomy, hearts, kind of what they do, things like that from lab. And I think you should be okay. Other parts of heart anatomy that we covered specifically in lecture, think about our intrinsic conduction system of the heart. So we remember how we talked how the SA node is the pacemaker, how that kind of sets the rhythm of the heart with some input from our autonomic nervous system as well. From the SA node, the signal to contract goes to the AV node, and then of course down the AV bundle, to the bundle branches, to the little Purkinje fibers, as we call them, and then that triggers ventricular contraction. Now, think about like what would happen if some of those things didn't function. So if our SA node didn't work anymore, would our heart still beat? What do you think? It would. Because remember, our SA node does intrinsically control conduction, but if it stops our AV node, also depolarizes at a specific rate. The problem with that is the AV node just does it too slow. But the AV node would actually become the pacemaker of the heart when if the SA node is damaged. It's just the AV node's just by itself too slow.
Would that
It
be
doesn't.
like an arrhythmia?
I'm sorry, Karina, you're echoing a lot. I can't really make out what you're saying.
Would that be an arrhythmia?
The echo is killing me. Type it in the chat. That's
He
what the
asked
chat's
if
for.
that would be an arrhythmia.
That would be an arrhythmia, exactly. So that absolutely would be an arrhythmia. So anything that's going to mess with the conduction system of the heart is going to cause an arrhythmia, like that.
I have a question real quick. So
Sure, Kate.
is that when, does the SA node fail and that's why people, and then the people have arrhythmia and that's why people get pacemakers placed inside them?
Absolutely. Yeah,
Okay.
and it's not just the SA node failing. It can be just like the SA node's working, but the conduction system between the SA node and the AV node isn't working. And that's what we call, remember we talked about heart blocks, that's a heart block, that sort of thing.
Thank you.
But I mean, your heart will still try to beat. As I said, if your SA node fails, the AV node will actually take over, but it tends to be just too slow. It beats at an intrinsic rate of about 40 beats per minute. So that's too slow for most people, even at rest, to keep them going, unless you have to be a really, really good endurance athlete. Let's see, anything else with heart stuff I want to think about? Let me see. Let's see, let's talk about blood. So I'll see if I can think of anything else with heart stuff as we're going along. I was just reviewing the exam today, so most of the stuff I remember to talk to you about. Blood components. So remember what blood is made of. So we have plasma, right? Which is mostly water, but it's got some electrolytes and nutrients and proteins in there as well. We've got erythrocytes, which are our red blood cells. They contain hemoglobin. Remember, that's our oxygen transport molecule, right? We have white blood cells. So it's not bad to just kind of review your white blood cells and a little bit of what they do. And then platelets. And remember, platelets are involved in clotting. So once again, just general functions of the blood components. Remember that like certain terms like hematocrit. Hematocrit is the percentage of red blood cells in whole blood. Usually it's about 45-ish%, and it kind of goes up or down depending on your gender. Blood vessels and their functions. So once again, this is a little bit kind of lab stuff coming out to play. So, and once again, you're gonna be taking your lab final before the lecture final, so that'll probably help to kind of think about that stuff. Think about like what specific blood vessels do. So for example, our superior vena cava brings deoxygenated blood back to the heart from the upper portions of our body. Our inferior vena cava brings deoxygenated blood back to our heart from our lower parts of our body. Our aorta.
Can you say that?
Oh, sure.
Thanks.
Sure. So our superior vena cava, for example, brings deoxygenated blood from like our upper parts of our body back to the heart, right? Our inferior vena cava brings deoxygenated blood from the lower parts of our body back to the heart, right? Our renal artery brings oxygenated blood rich in waste to our kidneys. Our renal vein brings deoxygenated blood, which is low in waste, out of our kidneys, because think about what our kidneys do, right? They cleanse the blood of waste. Our hepatic portal vein, right? Our hepatic portal system carries nutrient-rich blood from our digestive system to our liver to process nutrients. So just kind of like the major stuff, and like think about where it's going, where it's coming from, things like that, and they'll kind of give you an idea of like what sort of questions that might be asked. So like that kidney one's a good one. The hepatic portal system is always important. So those are things that you should really know for an exam like this. Let's see. Think about no, we'll get there. We'll get there. So that's blood vessels and their functions. That's good. The heartbeat cycle. All right. So we went over this pretty well when we did it in lecture, but it's been a while. So let's talk about it. So remember, we start with ventricular filling, right? Where all chambers of the heart are relaxed. The AV valves are open, right? That's our tricuspid and our bicuspid valves are open. And blood is literally flowing through our atria into our ventricles, and our ventricles start to fill up. They are topped off at the end of ventricular filling by the contraction of the atria. So remember, the atria contract, AV valves are still open, semilunar valves are shut, and that kind of tops off our ventricles. Now, our next part of our heartbeat cycle, remember, is isovolumetric contraction, which is a really, really fancy word to say that our ventricles start to contract, and they're contracting enough that our AV valves have slammed shut, but the pressure isn't high enough in the ventricles to open the semilunar valves. So it's like not even half a second. But it's called isovolumetric contraction. So our ventricles are contracting, the AV valves are shut, but they haven't started contracting enough to overcome the backward pressure from the blood in our major arteries pushing back on our semilunar valves. Okay. Then we have ventricular ejection, where the pressure in the ventricles is high enough that the semilunar valves open and we eject the blood out of the ventricles out into our pulmonary and systemic circulations. Then after that, we have, remember, isovolumetric relaxation, right? So during isovolumetric relaxation, our ventricles start to relax, the semilunar valves slam shut, but the pressure in the atria isn't high enough to cause the AV valves to open yet. And then finally, we get back to ventricular filling where the entire heart's relaxed, the AV valves are open, and everything's flowing in. So, okay. So think about like what valves are opening and what's happening. So when all of the chambers of the heart are relaxed, what valves would be open, gang, based on what we just said? So if our ventricles are relaxed, if our atria are relaxed, what valves are open and what valves are shut?
AV valves are open and the semilunar luminar ones are shut.
Exactly, Laura. Perfect. Exactly. So what's open and what's shut when our ventricles are actively contracting and blood is being ejected from the heart? So our ventricles are actively contracting, the blood is being ejected out of the ventricles. What valves are open and what valves are shut?
Semilunar are open and the AV valves are shut.
Perfect. If you can remember those two things, you're going to be fine for the exam. Okay. So keep that in mind. If you can remember that stuff, you're going to be good. All right. Heart tissue layers. So the heart itself, the wall of the heart has 3 layers, right? We have endocardium, which lines the chambers of the heart, which is nice and smooth, kind of simple squamous epithelial tissue, just there to reduce friction. We have our myocardium. Whenever you hear the word myo, gang, what should you be thinking?
Muscle.
Muscle, right? Just like myometrium is the muscular layer of the endometrium, myocardium is the muscular layer of the heart. So that's the actual contracting muscular part of the heart. And then we have our perimetrium, which is our outer serosal layer of the heart, which kind of covers the outer portion, which is technically the visceral pericardium as well. So those are our three tissue layers. So once again, endocardium, smooth, simple squamous epithelial tissue. We have our myocardium, which is the muscular contracting force, right? It's the muscular wall of the heart. And then we have the epicardium, which is kind of our outer layer, the serosal layer, which is our visceral pericardium as well. So remember those three layers, remember what they do, that sort of thing. Blood types. All right, so remember this all has to do with antigens and antibodies, right? So if I have type A blood, what antigen do I have on the surface of my red blood cells? Don't overthink it. If I'm type A, I have A antigen, right? That's what makes me A. All right. If I'm type B blood, what antigen do I have? Following that same logic.
B.
B. Exactly. If I'm type AB blood, what do I have?
AB.
A and B. Good. If I have type O blood, what do I have?
No antigen.
Right. No, at least not A or B antigen, because we could also remember, besides all that, we can be positive or negative, right? Right. So if I'm Rh positive, what extra antigen do I have?
D.
Yeah, D or Rh antigen. If I'm Rh negative, I just don't have that antigen. Right. So think about like blood types and blood transfusions. Remember, it's not just about the blood type, what antigens you have, it's also about what types of antibodies you produce in your blood. So if I'm type A, I'm going to make anti-B antigen in my blood. If I'm type B, I'm naturally going to make if I'm type A, I'm going to make anti-B antibody. Sorry, not antigen, antibody. If I'm type B, I'm going to make anti-A antibody. If I'm type AB, well, think about it, okay? Do we want to attack our own blood? No, agglutinate us and kill us, right? So if we're type AB blood, are we going to make any sort of antibodies for anti-A or anti-B?
No.
No, absolutely not. What if we're type O? What antibodies do you think we'll make if we're type O, if we don't have either A or B antigen?
A and B.
Yeah, anti-A and anti-B. We'll make anti-A and anti-B antibodies. Remember, you don't automatically make anti-Rh antibodies, but you will once you get first exposed. So we don't play games with that. We're going to make sure that if you're Rh negative, should you ever receive Rh positive blood? Absolutely not. So remember, Rh positive blood of the appropriate ABO type can receive Rh positive or Rh negative. Remember, Rh negative can only receive Rh negative blood. So think about a person who's like A negative, okay? Can you transfuse A negative blood to somebody with A negative blood? What do you think? Yes or no?
Yes.
What'd you say?
If you are A negative, can you transfuse blood to someone who is also A negative?
Yes.
Yes.
Yeah, you can always receive your own blood type. All right. If you are A negative, can you transfuse blood safely to someone who is AB positive?
No.
No.
Why not?
Wait, isn't the answer yes?
The answer is yes.
Because
why-
because you don't produce just because you don't produce the RH thing doesn't mean you can't transfuse blood to them. that they can't transfuse blood to you, but you can transfuse blood to them.
Right. And so think about it this way. So if I'm A negative, I just have A antigen on my blood. I'm transfusing it to somebody who is AB positive. So they already have the A antigen, right? Are they going to be sensitized to my blood? No. They're already Rh positive. So are they going to be sensitized to negative blood? No. There's literally nothing on my blood as an A negative person that AB positive person doesn't already have.
Is
And remember,
AB the universal receiver?
yes, AB positive is the universal receiver. Absolutely.
Again,
So
with-
anybody can receive AB positive. The big thing we as medical practitioners have to worry about with negative blood types is if a woman is Rh negative, what's she possibly at risk for? Anybody remember?
Having an Rh negative baby.
Well, not if she has an Rh negative baby, that's fine because she's Rh negative. She's not going to attack the baby if it's Rh negative.
Oh, it's
But-
positive.
Yeah. So does anybody remember what it's called if an Rh negative mom gets an Rh positive baby and then I remember the first one's going to be okay because but the second one's going to be in trouble because she could have erythroblastosis fetalis or hemolytic disease of the newborn. This was called. So only a woman who's Rh negative is even possibly at risk for that. If you're
But
Rh positive, you're fine.
I mean, it's academic, but my experience is that they just tested me automatically.
Yep, they do. And if
Automatic.
you're Rh negative, they just give you Rogam. So anytime anybody's a woman is Rh negative, they're just going to give you Rogam. But it's because you're at risk of perhaps developing hemolytic disease of the newborn or erythroblastosis fetalis, or your child getting that is why they screen you automatically and give you Rogam. It's because of that risk. Okay.
Thank you.
And I love erythroblastosis fetalis, so there's definitely going to be a question with blood types involving this on the exam. So keep that in mind. Let's see what else we got. Anything else with blood types?
What was the other name for it? Hemolytic, what?
Yeah, let me type it in the chat. So it's hemolytic disease of the newborn or erythroblastosis fetalis.
Sorry, I was just more likely to remember the other one.
Yeah, no worries. Which is why if I do put it on the test, notice me nodding my head, I'm going to have both as part of the question. Okay. So it'll literally have both terms. So you should remember at least one of them. Anything else on there? Let me think. Because we did blood types. We're pretty good with those. It's not a bad idea to just kind of review those, just be safe. Blood characteristics. That's gonna we pretty much went over those. Remember your white blood cells? So remember, white blood cells could be granulocytes or agranulocytes. The granulocytes have the granules in their cytoplasm. The agranulocytes don't. Remember, our granulocytes are like our neutrophils, our eosinophils and basophils. Our agranulocytes are the monocytes and the lymphocytes, things like that. But we'll talk about a little bit more of each of those in a little bit when we get to the immune system. Hemoglobin. So remember, what does hemoglobin do?
Transports iron.
Oxygen.
Oxygen, transports oxygen. It has iron, but that's how it transports the oxygen. So remember the main job of hemoglobin, it's got a big job. It transports oxygen. Remember, it also does transport a little bit of carbon dioxide. So it also does transport some carbon dioxide, but its main job is to transport oxygen. All right, the next one. This gave everybody trouble. on the exam when we did cardiovascular system. So there are, write this down, okay? There are 3 things that control blood pressure, okay? Star this. I don't want anybody getting this thing wrong because it's going to come up not just on this exam, but on NCLEX exams and things like that. 3 things control blood pressure. Cardiac output. So cardiac output, peripheral resistance, and number 3 is blood volume. So once again, the 3 factors controlling blood pressure are cardiac output, peripheral resistance, and blood volume. And just to kind of go over like what that stuff means, remember cardiac output is literally how much pressure your heart's putting out as it beats. So it's literally how hard is the heart beating. The harder it beats, the more pressure it's going to put downstream into the arteries. Peripheral resistance typically is the resistance inside the blood pressure, inside the blood vessels. And that's mainly controlled by the diameter of your blood vessels. The smaller the diameter, the greater the resistance. The bigger the diameter, the less resistance. So when they constrict, your blood pressure goes up. When they relax, your blood pressure goes down. And finally, blood volume. You try to pack more liquid into the tubes, it's going to increase the pressure. Okay, so as you put more fluids into the blood, pressure is going to increase. As you remove fluids from the blood, pressure is going to decrease. But remember those factors: cardiac output, peripheral resistance, and blood volume. Those are 3 factors controlling blood pressure. I don't want anybody to get that question wrong on this exam. Okay. All right, that brings us to heart sounds. So 2 main heart sounds. We have our lub, remember, which is our first heart sound. What's going on in our heart when we hear the lub? When we hear that first sound, what's slamming shut?
The valves.
Which valves?
AV.
The AV valve. So remember, lub is the AV valve slamming shut. The dup, which is our second sound, is the semilunar valve slamming shut. Okay. So once again, lub, first sound, AV valve slamming shut, dup, second sound, semilunar valves slamming shut. Okay. All right. Flow blood through the heart. So, once again, let's just kind of go through this nice and easy. Blood comes back to the right side of the heart from the systemic circulation. It's deoxygenated, flows into the right atrium through our vena cavas. That's right. Again, our superior and inferior vena cavas. Enters the right atrium. From our right atrium, it flows through the tricuspid valve into our right ventricle. From the right ventricle, well, you tell me, where does it go after it goes through the right ventricle? What valve does it go through? Remember, think of where our right side of our heart is pumping the blood to.
Pulmonary.
So yes, pulmonary valve, good, exactly, because I asked what valve, so it's going through the pulmonary valve. From our pulmonary valve, remember, it goes into our pulmonary trunk, and then to our pulmonary arteries, which brings the blood out to our lungs to pick up the oxygen. What's it come back to the heart through from the lungs?
Pulmonary
so
artery.
not pulmonary veins, because remember, veins always go back to the heart, right? So pulmonary veins back to the heart, then into our left atrium, and then from our left atrium through our mitral or bicuspid valve into our left ventricle. And then once again, our left ventricle is going to pump the blood out through our aortic valve or aortic semilunar valve into the aorta. And then once again, out through the rest of the body. So.
I don't know if this will be helpful to anyone, but it was helpful for me. I printed like a blank diagram off of Google and I filled it out and that helped me. So I don't know if
Mhm.
that's helpful to someone else.
Absolutely. It's extremely helpful. Did Professor Small give you like diagrams in lab that you could use?
Yeah, he did.
So use those.
Yeah,
Like kind of sketch it out on those, print it out,
in the
things like
PowerPoint.
that. Yeah. So in the PowerPoints, it's a good idea.
I actually drew the heart with the blood flow in it.
And I actually you can almost like draw it as like a heart because you can have like the upper parts as the atria and then the lower parts coming down to the point as the ventricles and just draw the line down between, separate it out. It's not too hard. So
you-
draw it out. If you can draw it, you're gonna remember it, is what it comes down to, gang.
Right.
Would you, whoever said that they Googled it, will you send the link to us? So we can,
Oh, the blank diagram.
yeah,
Yeah, I can do that.
thank you so much.
I mean, gang, I also have like blank diagrams from when I teach lab. I can post those if you'd like. I'm cool with it. So I do have some of those. And let me write this out quick. So I'll write it out in order. It's not bad to just kind of write this stuff over again. Once you are and remember the bicuspid is also called the mitral. I'll even say it's the left AV valve. There we go. So that's the entire list step by step. So you can sketch it right out without missing any of the steps.
Thank you.
Yeah, you're welcome. All right, so characteristics of arteries and veins. So that's our next part. So in general, veins have thinner walls, right? Veins have thinner walls. They have larger lumens and they have valves, right? Arteries are going to have thicker walls, smaller lumens, and no valves, right? So typically, like arteries are going to have thicker walls and veins of about the same division or same size. Yeah, that's kind of like what you kind of need to know about arteries and veins in general. Ooh, that brings us down to our ECG. All right, so remember our ECG, our P, our QRS, and our T waves. All right, so remember our P wave is atrial depolarization, right? Our QRS complex, or QRS wave, is ventricular depolarization. And then our T wave is ventricular repolarization. So once again, P, atrial depolarization. QRS complex. And then T is ventricular repolarization. All right, that's it for cardiovascular system. I think I've covered everything that we could possibly want to know about that. Lymphatic system. All right, so lymphatic organs, structures, functions, things like that. So what lymphatic organs are found in our pharynx here, gang?
Parathyroid.
The parathyroid gland is not a lymphatic structure.
Thymus.
Tonsils.
Oh, that's right.
Tonsils are in the pharynx. And remember what tonsils do. So they essentially screen anything coming in through our mouth and nose for like pathogens and stuff like that, right? So for antigen. Our spleen. What does our spleen do?
Kind of
Doesn't it filter?
It
filter.
filters. Yeah, it
Pathogen?
from what? What does it filter pathogens from?
The blood cells.
The blood, right? So it filters the blood. It filters the blood for pathogens. It looks for pathogens. It filters out old red blood cells and old platelets and things like that. Good, nice. What do the lymph nodes do?
Immunity.
Right, not quite. Remember, it's the cells that are the immunity. It's the lymph nodes that filter the lymph, right? Because one of our big lymphatic functions is like getting the excess fluid that's dropped off at our tissues back into our cardiovascular system. So like our lymph nodes filter the lymph. Other lymphatic structures, remember we have like our appendix and pairs patches in our intestines that make sure no pathogens breach the intestinal walls. We have MALT, which makes sure nothing penetrates our mucous membranes, or if things do penetrate the mucous membranes, they can deal with it. That sort of thing. So remember, the thymus, remember, is also technically a lymphatic structure, as well as a gland. Anybody remember what the thymus does? T for thymus, T for
T cells.
T cells, right? That's where the T cells go to mature to gain immunocompetence, things like that. Let's see what else. What are lymphatic system cell things like that? Lymph transport. Remember, lymph transport is always going to be slow. It's going to be low pressure. Very, very low pressure because it's very slow. Remember, it's powered by the contractions of adjacent tissues like pulsing veins, or I'm sorry, pulsing arteries or skeletal muscles moving. Remember, lymph vessels anastomose or interconnect very frequently, even more frequently than veins. And once again, our lymphatic vessels have valves to make sure that the lymph keeps flowing in one direction, just like veins. So keep that in mind. But once again, very, very low pressure system. Things move very slowly, kind of taking a meandering path, passing through the lymph nodes, so the lymph nodes can screen everything, all the lymph for not just pathogens, but cancer cells and debris and things like that. All right. Grab a quick sip of water here. That brings us to the immune system. So there's only like 2 questions on lymphatic system. Immune system, we have 12 questions though. Lines of defense. Okay, so we have first line of defense, second line of defense, and third line of defense. So first line of defense, remember, is chemical and physical barriers. Skin is part of your first line of defense. Mucous membranes are part of your first line of defense. Stomach acid. Believe it or not, it's part of your first line of defense. Tears and sweat and the chemicals therein are part of your first line of defense. Its job is to try to keep things out, keep things on the outside of you. All right. Second line of defense. All right. Cells and processes. All right. Second line of defense will include things like fever and inflammation. Fever and inflammation are second line of defense. Excuse me. Chemicals like interferons. So remember, interferons are antiviral proteins. They are released by virus-infected cells, and they tell other cells to up their defenses, but they also activate natural killer cells to come and kill infected cells. So interferons have all of those great effects against viruses. Complement, which enhances inflammation, increases phagocytosis, can actually literally drill holes into bacteria in a process called cytolysis, things like that. So that's, once again, second line of defense. Cells of second line of defense. Every other white blood cell except B cells and T cells are second line of defense.
What is it called? when
So
you drill holes? I'm sorry.
it's called cytolysis. Don't worry about it for the exam. I was just
Okay,
giving you
cool.
an
Thank you.
example of it. Okay. Worry about interferons. You notice interferons are on there specifically. You'll see. So once again, interferons, antiviral proteins released by virus-infected cells, warn other nearby cells to up their defenses against viruses, but also activate natural killer cells to kill virus-infected cells. Other parts of the second line of defense, so that would include our neutrophils that like to eat bacteria, our eosinophils that like to kill parasitic worms, basophils and mast cells which release histamine and heparin to promote inflammation. The natural killer cells, which will literally go against any virus-infected cell and a lot of tumor cells as well. Third line of defense, though, remember, is adaptive immunity. So these are our B and T cells. Okay. So remember, our B cells, once they get activated by their specific antigen, they turn into plasma cells. Remember, plasma cells make the antibodies. A lot of you got that one wrong on our immune system test, but write it down now. Plasma cells make antibodies. Okay, those are the cells that make the antibodies. Cytotoxic T cells kill virus-infected cells and tumor cells that display their specific antigen. So they're the virus and the tumor killers. Helper T cells are the bosses. Remember, they are required to activate the cytotoxic T cells. They help activate the B cells. Without helper T cells, you have no immune system, right? Helper T cells are like the generals of the army. Without them, you cut the head off you kill the general in the army, the entire army falls apart. In our case. So once again, helper T cells activate both branches of our adaptive immune system. The humoral branch, which is the antibodies from the B cells and the plasma cells. And then it's also the cellular branch with the cytotoxic T cells. We also have regulatory T cells, which, remember, kind of turn off the immune response once everything is tamped down. They also may help prevent autoimmune diseases. That's kind of what we're working on to try to cure autoimmune diseases right now. All right. Those are our three lines of defense. And we'll go over a couple more of that stuff as we go further down through our immune system. Passive versus active immunity. So remember, passive immunity, you are not activating your own third line of defense. You're not activating adaptive immunity. You are not making your own antibodies. You are not making memory cells to remember anything. Passive immunity, you are receiving antibodies from somebody else. So it could be naturally acquired through the mom through the placenta or through breast milk. It could be artificially acquired from getting injections of antibodies from somebody else who's already like suffered through a disease or had a vaccine or things like that. Active immunity, once again, you are activating your adaptive immune response. You are generating your own antibodies. You are making memory cells. It is long-lasting protection. And there's two ways we can get it. We can have naturally acquired active immunity, which I'm developing right now to this cold virus I have. I was infected with the cold virus. I have gotten sick. At this point, I am hopefully making my own antibodies and in this case, cytotoxic T cells and memory cytotoxic T cells that will hopefully protect me against this particular strain of virus. And then finally, we have artificially acquired active immunity, which is vaccines. So we receive either killed forms of the pathogen or killed forms of the virus that activates our adaptive immune response. And once again, we make our own antibodies, we make our own memory cells, and it's once again long-lasting protection. Let's see. Parts of the innate defense system, we kind of did that because remember the innate defense system is our first two lines of defense. So once again, innate defense system will be the chemical and physical barriers, the skin, the mucous membranes, the tears, the sweat, things like that. It's going to be inflammation. It's going to be fever. It's going to be our neutrophils and our basophils and our eosinophils and our macrophages and our dendritic cells and all that good stuff. Let's see. Well, remember, any sort of anything from the adaptive immune system is not going to be innate, right? So remember, innate is always works all the time against every type of antigen. The adaptive immune response targets specific antigens once it's back, once it's activated. Let's see. Talked about second line of defense. Inflammation is part of our second line of defense, right? Remember our cardinal signs of inflammation: redness, heat, swelling, pain, and loss of function quite often. Interferons, once again, antiviral proteins released by virus-infected cells, tell other cells to up their defenses, and also activate natural killer cells to kill the virus-infected cells. So that's, they do a lot. They're good. Let's talk a little bit more about the inflammatory response. So things that happen during the inflammatory response. So remember, as soon as anything damages your tissues, they're going to release chemicals that cause the inflammatory responses. These chemicals are going to cause first your capillaries to dilate, which is going to increase blood flow. That causes the redness and the heat. Clotting factors are going to infiltrate the tissues, which are going to prevent blood loss, but also going to kind of wall off any pathogens that might have snuck in as well. The fluids will leak into the surrounding tissues because the blood vessels get leaky, causing the swelling. And then the pressure of the fluids and prostaglandins being released by the damaged tissues and kinins and bacterial toxins are going to cause the pain. So once again, increased blood flow, blood vessels become leaky, redness and heat and swelling. The pain is going to be caused by the prostaglandins and the bacterial toxins and things like that. Once again, clotting factors are going to help things be walled off. So just kind of review those things because they're all part of the inflammatory response. Yeah, cool. All right. Humoral immunity. So humoral immunity, remember, is what works best against pathogens and antigens outside your cells. So in this case, with humoral immunity, your B cell gets activated. The B cells transition into plasma cells and memory cells. The plasma cells are effector cells. They're short-lived, but what they do is they pump out antibodies and more antibodies until they die. The memory cells stick around once again. And once again, you have more memory cells, so they're going to be kind of stationed throughout your body at this time after they're made. And they're just going to watch for that particular antigen to come back. And that's the basis of immune system memory. So remember, the first time your immune system comes into contact with an antigen, the antigen challenge, so to speak, which usually occurs in your lymph nodes, or somewhere in your lymphatic tissues. It's going to activate the adaptive immune response. We're going to start making our plasma cells. We're going to start making our memory cells, things like that. The first response is going to be kind of slow, and it's going to get rid of stuff, but it's going to take a while. That's why, like, when this cold thing hits me, it's going to take like a week or so to get over it. Excuse me. If that's the primary response, if that antigen comes back, we're going to mount a secondary response. Anybody remember the characteristics of the secondary immune response?
Fevers.
Well, we might not even have a fever. So remember with the secondary immune response, because I mean, fever is just anytime it gets triggered during our any immune response. It doesn't even have to go all the way to the adaptive immune response. Secondary immune response is when an antigen comes back and it triggers those memory cells.
It gets marked by like a B cell.
You're still thinking primary. So secondary immune response, remember, is the second time you get it. The memory cells find that antigen a lot faster. And the thing about the secondary response, and the thing I really want you to remember, gang, is that it is extremely rapid and extremely strong.
And it's
so the second
memory cells.
Yeah, it's because of the memory cells. It's the memory cells detect the antigen's return much more quickly than what we saw with the primary response. And they mount an extremely rapid and overwhelming response, so much so that most of the time you're not even going to have symptoms. And I see Jennifer's dog, and that is awesome. I love it. All right. So that's immune system memory. So remember, it's the memory cells stick around, and essentially they're waiting behind the door with a baseball bat. And as soon as that antigen rears its ugly head again, they just whack it over the head as soon as it steps through the door. Super rapid, super overwhelming response. And you have much higher levels of antibodies that last a whole lot longer. So it's kind of got like it's got everything going for it when it comes to a response to an antigen. The secondary response is just faster, more overwhelming, many more antibodies, and they just stick around a whole lot longer. And it's just better for everyone and everything. Let's see. Cells of immunity. So we've talked about them, but let's go over our adaptive immune cells again. So once again, helper T cells are the bosses of the adaptive immune response. Helper T cells have to help activate the B cells. They have to help activate the cytotoxic T cells. Without the helper T cells, we don't really have an immune response. Okay. B cells, once they're activated, some of them become plasma cells. Plasma cells make the antibodies. Some of them become memory B cells, and once again, they stick around for the secondary response. Cytotoxic T cells, once they get activated, they go out and kill virus-infected and tumor cells, displaying their specific antigen. But once again, some of them also become memory cells that if that antigen ever shows its ugly head again, they're going to mount, once again, a much more rapid secondary immune response to those. Suppressor T cells, remember, suppressor T cells, the only thing they do is they turn off the immune response. Once the immune response is essentially, once the pathogen has been cleared or once the antigen has been cleared, and they may have a part in preventing autoimmune diseases. We're not really sure. We hope that works because if we can activate them, we might be able to cure some autoimmune diseases. All right. That brings us to respiratory system. So this is all done. Let's highlight all those since we're good with that. All right, that brings us to the respiratory system. So what's the main function of the respiratory system, gang? Two
Exchange.
words. Gas exchange. Exactly. If you remember that, you're going to be happy. And so am I. Respiratory system anatomy. Remember the parts of the respiratory system and a little bit of their functions. So like our nasal cavity is kind of the entrance. It warms, moistens, and filters the air. Followed by that is our pharynx. Remember our pharynx. I love the pharynx because it is not just a respiratory organ, it is a digestive organ as well. So remember it's our essentially crossing over point for both our respiratory and our digestive systems. Our next respiratory organ, which is respiratory only, is our larynx. So remember our larynx provides a patent airway, but it's also our voice box. It's where our vocal folds are for sound production. Underneath our larynx, we have our what? Anybody remember? This is good for lab too. So trachea, right? So our trachea. Remember the structures of the trachea, especially the tissues. So remember our trachea consists of kind of a hodgepodge. So we have pseudostratified ciliated columnar epithelial tissue, which makes up the mucosal layer. We have hyaline cartilage, which makes up the C-shaped rings of the trachea. We have smooth muscle, which forms the trachealis at the back of the trachea, connecting the two ends of the C. And then we have a fibrous adventitia that kind of covers everything over. That's actually not a bad thing to remember for the lab final too, because like a lot of times there's a slide showing exactly those tissues, and you see all of those tissues. It's got to be the trachea. So once again, tracheal tissues in particular, pseudostratified ciliated columnar epithelium, smooth muscle a little bit, hyaline cartilage, and then a fibrous adventitia is always important in there too. Other respiratory tissues, remember, as we go further and further down through our respiratory tree, we start losing cartilage and we start gaining more and more smooth muscle. The epithelial tissue also starts getting thinner. So by the time we get to our respiratory surfaces, we have mostly simple squamous epithelial tissue in the alveoli themselves. Also inside the alveoli though, remember we have alveolar macrophages, which kind of keep things clean. But we have also one of my other favorite cells, which is our type 2 alveolar cells that secrete something very, very important, which is called surfactant. So remember, surfactant reduces surface tension in the alveoli to keep the alveoli from collapsing. So without surfactant, remember, gang, all of our lungs would be collapsed right now. So thank evolution for surfactant. So once again, surfactant reduces surface tension in the alveoli to prevent lung collapse. Without it, none of us would be here. Let's see. Other respiratory tissues. No, I think we're pretty good with that. I think we're good. Carbon dioxide transport is our next one. All right, so the bulk of carbon dioxide is transported, remember, as bicarbonate ion. Right? We have some transported on hemoglobin as carbaminohemoglobin. We have a tiny bit that's actually dissolved CO2 in the plasma. But the vast majority of carbon dioxide is transported as bicarbonate ion. How do we get from carbon dioxide to bicarbonate ion? So remember, there is an enzyme. There is an enzyme inside our red blood cells called carbonic anhydrase that converts carbon dioxide and water into hydrogen ions and bicarbonate ion. All right, that's carbonic anhydrase's job. I'm not going to make you know the chloride shift, but I want you to know carbonic anhydrase. So once again, carbonic anhydrase inside our red blood cells, inside our erythrocytes, converts carbon dioxide and water into hydrogen ions and bicarbonate ion. And remember, that is how we transport the bulk of the carbon dioxide in our blood. Okay. Remember that. All right. Inspiration and expiration, particularly muscles of inspiration and expiration. So quiet inspiration. Quiet inspiration. We're breathing in. What muscles are contracting? Anybody remember?
External.
Yes, external intercostals. And don't forget our main muscle of respiration, which is the what? The diaphragm. So remember, quiet inspiration, external intercostals, and the diaphragm are contracting. Okay. Quiet expiration. What's contracting with quiet expiration?
Nothing.
Nothing, exactly. It's just muscles relaxing, right? That's exhalation. That's expiration, right? Diaphragm relaxes, the external intercostals relax, and we expire or we exhale, right? Let's see. What else we got? Forced expiration. We're doing forced expiration. We do have muscles contracting. So what muscles are contracting with forced expiration? Anybody remember?
External.
Isn't like your
External and internal.
internal,
Just the internal intercostals,
Costal.
mhm.
but also abdominal muscles. Okay. Those are the two main ones that would show up on the test. So if we're doing forced expiration, internal intercostals and our abdominals. Forced inspiration. So like if you have COPD, we're going to be using our kind of our chest muscles. So we're going to be using our neck muscles. So our scalenes, sternocleidomastoids, pectoralis minor, things like that. All right. That brings us to pleural membranes. All right, pleural membranes. So these are membranes that cover our lungs and line the thoracic cavity. Oh, yeah, it is about that time. So, all right, I'm gonna keep going. This is being recorded, so I'm gonna keep going until this thing's done. All right. Stick around if you want, if you think you're gonna have questions, but if not, go to bed. It is late.
Have a good night.
All right.
Professor, thank you.
You're welcome. I will see you all next Thursday for the final. And if you have questions before then, email me, or if you want to make an appointment to like talk to me on Zoom, we can do that.
And can I ask a quick question right now? Is it in person?
No, it's remote.
No.
So I
Oh.
changed it to completely remote. So the
Okay,
final
I was
exam
under the
is remote.
assumption we had to come in.
No, you don't. No, I changed. That was the original plan, but like a lot of you just couldn't make it. So 125 questions on the final, Miriam. All multiple choice. So 100% multiple choice. And I said, listen to this recording. Because as I said, I will go through all the rest of the systems. Make sure you listen to it. Take notes, review it.
Okay. Thank you.
You're welcome, gang. Have a good night.
Thank you. Good night.
All right. See y'all next week, if not sooner.
All right.
All right, so pleural membranes. So remember, we have two pleural membranes. We have our visceral pleura, which cover the actual surface of the lungs, the organs themselves. And then we have the parietal pleura, which lines the actual thoracic cavity. It's the epithelial lining that lines the surface of the thoracic cavity surrounding the lungs is the parietal pleura. That brings us to Henry's, Boyle's, and Dalton's laws. worry about Boyle's law, gang. Remember Boyle's law is that once again, pressure and volume are inversely related. So as the volume increases, pressure decreases. As volume decreases, pressure increases. Okay. So keep that in mind. So as I said, focus on Boyle's law. Henry's and Dalton's are a little bit too niche to worry about for the final, but Boyle's is definitely important because that's how we inhale and exhale. So once again, Boyle's law, as volume increases, pressure decreases, and as volume decreases, pressure increases. All right, that brings us to pH. So this is kind of important, and this actually goes a little bit to the lab stuff. Hey, Kate, everybody else left because it's like 9 o'clock, so stick around if you want.
I'm gonna go too because I've been like studying all day long and working at the same time. So.
Get some sleep. All right.
Okay.
Have a good night.
Bye.
All right. Good night.
Good night. Thank you.
Oh, my pleasure. All right, so pH. So things to remember about pH and respiratory system. So remember, pH is directly related to carbon dioxide levels. Okay. So as carbon dioxide levels increase, pH decreases, right? As carbon dioxide levels decrease, pH is going to increase. So that's kind of the big things I want you to remember about pH in the respiratory system. So once again, pH is related to carbon dioxide levels. So as carbon dioxide levels increase, or concentrations of carbon dioxide levels increase, pH is going to decrease. As carbon dioxide levels decrease, pH is going to increase. And once again, that's all has to do with this stuff. Think about like where that's going to happen, stuff like that. Gas exchange. Remember what drives all gas exchange. It's diffusion. So remember, diffusion is what drives all gas exchange. Diffusion is the type of molecular movement that drives gas exchange everywhere in your body, in the lungs, in the tissues, you name it, gas exchange. Gas exchange is what's driving it. All right, controls of breathing rate. So remember, there's two things that control respiratory rate. Okay. Really important. There's two things that control respiratory rate. pH and carbon dioxide levels, which are related. Remember, oxygen levels don't really factor in very much. So if carbon dioxide levels go up, our pH is going to go down, and that's going to increase respiratory rate. So either carbon dioxide levels going up or pH going down is going to increase respiratory rate. Carbon dioxide levels going down or pH going up is going to decrease the respiratory rate. So once again, if pH is too low, respiratory rate is going to increase. If pH is too high, respiratory rate is going to decrease. If CO2 levels are too low, CO2 levels are too low, respiratory rate is going to decrease. If CO2 levels are too high, respiratory rate is going to increase. So it's kind of opposite for CO2 and pH, but once again, CO2 and pH, the levels of CO2 has the opposite effect on pH. So keep that in mind. So that's just to make sure we're all on the same page. So as CO2 levels go up, respiratory rate increases. As CO2 levels go down, respiratory rate decreases. As pH goes down, respiratory rate increases. As pH goes up, respiratory rate decreases. Okay, remember that. And you know, we just did muscles of inspiration and expiration, so I think we're good with that. So we will skip over that, so I can actually go to bed at a decent hour tonight. All right, that brings us to the digestive system. So there's 14 questions on that. So peristalsis versus segmentation. Peristalsis, remember, one-way movement, straight shot through the digestive system caused by rhythmic muscular contractions, but it's going one way from mouth to anus. Segmentation is different. Segmentation occurs only in the small intestine, whereas peristalsis goes through like the esophagus and the stomach and the small intestines and large intestines all the way out through the anus. Segmentation only happens in the small intestine. It is the two-way back and forth movement of food in the small intestine for both mixing and mechanical breakdown purposes. Right, so it's part of mechanical digestion and mixing. So it's a back and forth movement, two ways. That's segmentation. Digestive system organs and their parts and their functions. So that's a lot of stuff, gang. So we'll start at the beginning. So remember, we have our mouth, which has salivary glands in it. It has teeth in it, has the tongue in it. So remember, our mouth is for ingestion. It does mechanical digestion by chewing the food, breaking it down physically into smaller pieces. But it also does chemical digestion through salivary amylase. Remember, amylases are enzyme that breaks down starches and other carbohydrates into simpler forms. Remember the digestive secretion chart that we worked on and I posted? You should have it filled out. Look over that thing. Okay, it's gonna help. From our mouth, we're heading into our pharynx. Once again, our common passageway for food and air. So pharynx is both part of the digestive and respiratory system. From there, our next part of our digestive system is esophagus. Its only job is propulsion. It's just moving, or it's motility. It's moving things through down toward the stomach. From the pharynx to the stomach is its job. It's lots of mucus glands to keep things lubricated, things like that. Now our stomach, remember our stomach's job, it stores the food for a little bit. It does mechanical breakdown through the churning and the mixing, or mechanical digestion through churning and mixing, but it's also where chemical digestion of proteins and lipids starts. So proteins, pepsin, lipids, we have gastric lipase, but also salivary lipase doesn't start working until it's in the stomach because it needs the low pH. Remember the cells and what they secrete of the stomach. So remember, chief cells. This is something that gave y'all trouble on the digestive system test. Chief cells are chiefly for enzymes, right? Which means chief cells secrete the pepsin, which digests proteins. It secretes the gastric lipase, which digests fats. Parietal cells make hydrochloric acid and intrinsic factor. And remember what intrinsic factor is used for. Intrinsic factor allows you to absorb B vitamins so you can make red blood cells. After that, if we're going to stay in just our gastrointestinal tract, we're going to go into our small intestine. Remember the functions in the small intestine: absorption and chemical digestion. Now, most of the chemical digestion is because of our pancreatic enzymes and then the brush border enzymes, which break down all of our nutrients into their final absorbable forms. As I said, the vast majority of absorption happens in our small intestine. Remember, it's got special adaptations, special features that increase its surface area for absorption. These include villi, finger-like things, the circular folds, which cause the chyme to spiral through and slow it down. And then we have the microvilli on top of the villi that increase the surface area even more. After that, we have our large intestine. Remember, the only thing that's going on in our large intestine is that we're absorbing water and some vitamins that are provided by our bacterial flora. We're also making gas and compacting the feces into kind of more solid stuff. And then it's eliminated through our anus. Remember, it's stored in our rectum for a little bit. Once our rectum gets so full, we get the urge to defecate, and it's eliminated through the large intestine. Don't forget the accessory organs. Okay. Salivary glands, once again, make saliva, which contains salivary amylase, excuse me, which breaks down starches in our mouth. We have our pancreas, which produces a whole slew of enzymes. Trypsin, the enzymes of the pancreas include trypsin, peptidase, and carboxypeptidase, which all break down proteins. We have pancreatic amylase, which, as an amylase, remember, amylase breaks down carbohydrates, starch, things like that. And then it has pancreatic lipase, which chemically breaks down fats into fatty acids and glycerol and monoglycerides and things like that that we can absorb through our small intestine. Remember, the pancreas secretes its pancreatic juices through ducts into the duodenum, the very first loop of the small intestine. We also have our gallbladder. Remember, our gallbladder stores bile, which is then secreted into the small intestine. And remember, the bile emulsifies fats. So that's our gallbladder that stores bile for the emulsification of fats. Oh, we need to talk about the parts. I forgot about the parts. Stomach parts, fundus, which is the dome part. The body, the pylorus, which kind of squeezes down toward the pyloric sphincter toward our small intestine. Small intestine has 3 segments. First segment's the duodenum, which is really short. Then we have our jejunum, where most of the chemical digestion and absorption takes place. And the last part is the ileum, which leads into our large intestine at the ileocecal valve. So remember, our 3 parts of our small intestine: duodenum, jejunum, and ileum. The ileum is the 3rd part, the last part. Our large intestine has its own segments. So we start with our cecum, which is our first section where what's left of our undigested food heads into after it gets expelled from our small intestine. Right hanging off of our cecum is our little vermiform appendix. Then we go into our ascending colon, then our transverse colon, and then our descending colon, then our sigmoid colon, then our rectum, and then our anus. So those are all important parts. Let's see. Talked about the enzymes in the pancreas. Hormones for the digestive system. This is something also that gave some of you trouble. One of the main, most important hormones is gastrin. Remember, gastrin is a hormone secreted by our stomach that increases gastric juice secretion and gastric motility, so stomach movement. So remember, it increases secretion of pepsin and hydrochloric acid and gastric lipase, and also increases stomach movements. So that's gastrin. Remember, secretin and CCK reduce that, right? Remember, gastrin increases gastric, increases gastrin, increases gastric juice secretion and things like that. Digestive enzymes and their secretion and digestive secretions. Once again, look over your sheet. Make sure you look over the sheet we took time to kind of fill out when we did digestion. What happens to food as it's digested? So this is actually just something nice to kind of go over. So let's see. Oh, other parts. Rugae. Don't forget rugae are in the stomach, which increases, well, they're kind of like folds that allow the stomach to expand. Let's talk about what happens to food when it's eaten. So let's say we start eating a hamburger. So a hamburger's got fats in it, it's got protein, it's got carbohydrates from the bun. So as soon as we start chewing that thing in our mouth, we're mechanically digesting it, breaking it down. The salivary amylase, once again, amylase breaks down starches, starts breaking down the starch in the bun. and then we swallow it. The food's going to travel down our esophagus via peristalsis and get to our stomach, where it's going to mix with the gastric juices. As food enters our stomach, we're going to see an increase in the hormone gastrin secretion, which is going to increase gastric juice secretion and gastric movements. The proteins are going to start being broken down by the enzyme pepsin. And the fats are going to start broken down by gastric lipase and lingual lipase from the mouth. A little bit of chyme is going to start to be squirted through our pyloric sphincter into the duodenum of our small intestine. That's going to essentially kick off our intestinal phase, where we're going to start secreting our pancreatic juices. We're going to start secreting bile. Bile is going to emulsify the fats, kind of break them into smaller pieces. So pancreatic lipase can actually break them down chemically. It's at this point that salivary amylase will also, or sorry, pancreatic amylase will continue breaking down any starches that were broken down in the mouth. Trypsin, carboxypeptidase, and chymotrypsin will start breaking down from the pancreas. Will start breaking down the proteins even more, and then the brush border enzymes in the small intestine will finish breaking down the proteins and the carbohydrates into their absorbable forms, which are going to be amino acids and simple sugars. Lipase, pancreatic lipase takes care of all the fat stuff to break it down to fatty acids and monoglycerides and glycerol, which are going to get absorbed and then repackaged as triglycerides and then put into our lymphatic system through the lacteals. Anything left over is going to make its way down the small intestine through the jejunum and into the ileum, and then into the large intestine, where our bacterial flora will start eating our leftovers and giving us vitamins in return. Water and those vitamins is going to be absorbed by our large intestine, and feces is going to be compacted, and that's going to be eliminated through our anus. And that's kind of what happens anytime you eat a meal, which is kind of cool. That brings us to nutrition. All right, so insulin, glucagon, and epinephrine. So we eat a large meal. This is our fed state. So where we talked about our fed state. Insulin is going to be secreted. So insulin does a lot. So insulin is going to tell our fat cells and our muscle cells and other body cells to start increasing glucose production or glucose absorption, but also increase absorption of amino acids. That's going to tell our cells to actually store these things or make use of them. So it's going to increase protein synthesis, stuff like that. During our fasting state, we're going to start like about 3 or 4 hours after we ate our meal. And once food is cleared out of our digestive system, we'll enter our fasting state. Glucagon is going to be released, which is going to increase blood sugar levels. It's going to tell our fat cells to kind of start breaking things down to release this blood sugar. It's going to tell our liver to break down glycogen and make them, oh, hold on, it's St. Nicholas Day, so the kids need their shoes so they can put them out for St. Nicholas to put things in. So let me just go open. I'm going to pause the recording while they do that. So let me, give me a second, gang, I'm sorry. Hey gang, I'm gonna go put the kids to bed quick, and I will be back to continue recording in a minute. All right, so I'm back. All right, so essential nutrients. So remember, these are the nutrients that we have to get from our diet. Mainly because we can't get them any other way. We can't make them in our liver. So remember, essential nutrients are essentially the nutrients that can't be made in our bodies, so they have to be consumed in our actual diet. Remember, metabolism was a part of all this. So it's nutrition and metabolism. Remember, after we eat a meal, our metabolic rate tends to go up because it takes a lot of energy to like break down all of those nutrients and then also absorb them and transport them to our liver and process them and things like that. So we talked about insulin and glucagon. Um, remember the processes for making ATP? So glycolysis, intermediate phase, citric acid, oxidative phosphorylation. Remember they all um, essentially make ATP for us. So keep that in mind. Let's see. Remember amino acid metabolism? Remember as amino acids get broken down and recycled, they get deaminated because the ammonia is actually toxic. And remember, the ammonia gets converted into urea, which gets excreted through our urine. Let's see what else is going on. End products. So reactants of all our phases. Remember, so it starts with a molecule of glucose typically. Goes through glycolysis. It gets broken down to 2-pyruvate. We get 2 ATP out of that. The pyruvate gets sent in. Oh, and also like FADH. The pyruvate gets tossed into the intermediate phase, which converts pyruvate to citric acid. Citric acid cycle goes through with creates more ATP. This is also where carbon dioxide is produced in both the intermediate phase and the citric acid cycle. The NADH and the FADH that's been produced so far gets tossed into oxidative phosphorylation, where it reacts with oxygen and water. So it reacts with oxygen to make water. I'm sorry, is our product. Remember, oxidative phosphorylation, electron transport chain, makes the bulk of the ATP. If there's about 30 total ATP meta this entire process, like 28 or so is actually ends up being made in oxidative phosphorylation. So keep that in mind. So remember, end products of aerobic respiration, total, that entire thing, we get carbon dioxide, water, and about 30 ATP is the end products. As I said, the reactants is oxygen and glucose is what it comes down to. So keep that stuff in mind. I said there's not a lot of questions, but they're a little bit specific. So once again, ammonia being released by the breakdown of amino acids gets converted into urea in a process called deamination. Remember, insulin's released, which causes the uptake of glucose and the processing and storage of nutrients and the utilization of nutrients. Essential nutrients have to be gotten through our diet, things like that. Remember, when we eat, technically our metabolic rate goes up because we're using a lot of energy to break down and store and process all those food molecules and things like that. All right, so that brings us to our urinary system. Remember that, like, just the major functions in the urinary system, we're trying to get rid of wastes. It's kind of the main function. We're trying to get rid of metabolic wastes. But besides that, or getting rid of nitrogenous wastes like urea. Besides that, though, we're also trying to maintain the saltwater balance, maintain the acid base balance as well, because the urinary system plays a big part of that. Remember that there are different parts in the functional anatomy. So remember, we have our glomerulus and the proximal convoluted tubules and our distal convoluted tubules and the loop of Henle. I remember kind of the general anatomy. So right, like the most of the stuff's in the renal cortex. So there are our glomerulus and our proximal convoluted tubule and our distal convoluted tubule. They're all located mostly up in our renal cortex, the cortex of the kidney. Remember, the loops of Henle dip down into the renal medulla and come back to form our distal convoluted tubule, and then we head down into our collecting ducts, which once again head down into our renal medulla. through our pyramids. All right. Proximal convoluted tubule itself, remember, has a brush border of microvilli. It's simple cuboidal epithelium. Remember that helps with the tubular reabsorption. Greater surface area for tubular reabsorption, things like that. Glomerulus itself is remember the kind of tuft of capillaries that does the filtration. It's optimized for filtration. It's fenestrated capillaries, right? So it's there for filtration itself. What else we got? Talked about functions. Remember the different types of nephrons, speaking of anatomy. So we have cortical nephrons and juxtamedullary nephrons. Remember, cortical nephrons make dilute urine. Juxtamedullary nephrons make concentrated urine. Remember, they're the ones with the really long loops of Henle, so we can absorb more water and salt from the urine. Remember ADH and aldosterone. That's an important one. So remember, ADH. This kind of goes back to endocrine system too. Remember, it's released from our posterior pituitary. It acts in the collecting ducts by inserting aquaporins, so more water is reabsorbed. At really, really high levels, it causes vasoconstriction. Mainly, it's released when we are dehydrated. So it actually increases in levels when we're dehydrated. So it's kind of our dehydration hormone. Aldosterone, on the other hand, kind of does similar things. It also acts in the collecting ducts, but what it does is it increases sodium reabsorption, but it also increases water reabsorption. And doing both of those things, if you remember, actually also increases blood pressure. So aldosterone is one of those things that by increasing salt and water reabsorption is going to increase blood pressure as well. Remember how the kidneys kind of deal with pH. So remember, if we are in an acidosis state, the kidneys, if our blood gets too acidic, the kidneys will respond by secreting hydrogen ions and retaining bicarbonate ions. And that's going to raise pH. It can also do the opposite. If we're in alkalosis, we can actually do the opposite. We can, if our blood pH gets too high, we're going to excrete bicarbonate ion and retain or hold onto hydrogen ions. So keep that in mind. Let's see, things that might increase urine output. So things like, things that would decrease urine output. So ADH, like dehydration, low blood pressure, because that's going to decrease GFR, internal bleeding. These are all things that would actually end up decreasing urine output. Let's see. All right. Oh yeah. So acidosis and alkalosis. So this is more of a lab thing, but this is kind of a crossover question from lab. So we have respiratory acidosis and alkalosis, which is something going on in our respiratory system is altering the pH by increasing or decreasing CO2 levels. So if we have a respiratory issue like COPD or something like that, we're not going to be able to exhale enough CO2. And that is going to cause more CO2 to be held onto. That's going to lower blood pH. So that would be a cause of respiratory acidosis. Respiratory alkalosis is if we're actually exhaling too much CO2. So like if we're hyperventilating, that would be respiratory alkalosis because we're getting rid of too much CO2 and that's actually raising our blood pH. Metabolic acidosis would be something that would, something going on metabolically that would increase or decrease the pH of our blood. Alcoholism can do that. Things that would increase, like things that would cause metabolic alkalosis would be like extreme vomiting because we're getting rid of so much stomach acid. We're trying to replace it with hydrogen ions from our blood, and that would do that. But respiratory causes are much more common. So once again, hyperventilation is going to cause respiratory alkalosis, COPD, or other breathing issues we call respiratory acidosis. So once again, and once again, our kidneys will try to compensate for those by if we're in acidosis, we're going to try to excrete hydrogen ions and hold on to bicarbonate ions. If we're alkalosis, our pH is too high, we're going to excrete bicarbonate ions and try to hold on to hydrogen ions. Remember bicarbonate buffer system, bicarbonate ions are essentially our buffer in our blood. It's used to transport CO2, but remember it also acts as a buffer resisting changes in pH. So it helps maintain the acid base balance. So it's a multitasker, it's like to say. All right. That brings us finally. Oh yeah, loop of Henle. So remember loop of Henle. Once it dips down into the cortex. The descending or thin limb is where we reabsorb water. The ascending or thick limb is where we reabsorb salt. Remember that's always going to happen in the loop of Henle. Remember it's really long in our juxtamedullary nephron, so it makes more concentrated urine. Anything else on the kidneys? No, just kind of double check everything on that. All right, so oogenesis. So that's making it. It's ladies making eggs. So remember, oogenesis starts in fetal development with the oogonia making primary oocytes. The primary oocytes start meiosis 1 and then pause in the midst of meiosis 1 until puberty, where one primary oocyte gets selected to complete meiosis 1 and become a secondary oocyte. The secondary oocyte is what actually gets ovulated. The secondary oocyte may never actually complete meiosis 1. Because to complete meiosis 1, the secondary oocyte must be fertilized by a sperm. If it is fertilized by a sperm, it immediately, completes meiosis 2. So once again, the secondary oocyte has completed meiosis 1. It is now haploid. It's not yet completed meiosis 2. Secondary, excuse me, gang, I think my cold medicine's wearing off. Yeah, it is. Secondary oocyte only will complete meiosis 2 if it is fertilized. As soon as a sperm penetrates the oocyte, it's going to complete meiosis 2, become an ovum, and then immediately becomes a zygote as the two nuclei fuse. So remember, most of the time, oogenesis doesn't actually make it all the way to an ovum. It's only if it's contacted by a sperm. But once again, it starts in fetal development, but it doesn't end until except once a month typically once a month after puberty. All right, the follicular cycle. So remember, we have 3 phases of the follicular cycle. So our first phase is the follicular phase. The second phase is ovulation, and the third phase is the luteal phase, where we're actually secreting estrogen and progesterone from our corpus luteum. So remember, after ovulation, the leftover thecal and granulosa cells of the follicle kind of come together and form the corpus luteum, like kind of a yellow glandular tissue inside the ovary. It's going to stick around for a little while to secrete estrogen and progesterone. Remember, the estrogen and progesterone maintain the functionality of the functional layer of the endometrium to make sure that it is in its most receptive state if there is a fertilization event and an embryo tries to implant. If there is no pregnancy, the corpus luteum degenerates within 10 days to the corpus albicans, and the ovarian cycles Our follicular cycle starts all over again. All right, next one is structures of fetal circulation. So once again, it's I hope something that you kind of went over in lab, but if not, so remember the fetal circulation has blood vessels and features that adult circulation doesn't have. In the heart itself, there's two things. We have the ductus arteriosus, which is kind of bypass for the pulmonary circulation during fetal circulation because you're not breathing. So that's actually a little blood vessel right in between the pulmonary trunk and the aortic arch. It closes at birth. Excuse me. You also have something called the foramen ovale, which becomes the fossa ovalis, if you remember from the heart. The foramen ovale is, once again, a bypass going between the left and right atria. Once again, allows the blood to bypass the pulmonary circulation because we're not using it. As we go further down, we have our ductus venosus, which goes through the liver. And once again, it allows kind of the liver to be bypassed because once again, the mom has already processed all the nutrients for you. You don't really need to do any nutrient processing in there. And leading into the ductus venosus, coming out of the umbilical cord is our umbilical vein. And once again, the umbilical vein is bringing oxygenated blood from mom into the fetus. Coming off of our inferior vena cava, we have our umbilical arteries. These carry, actually coming off the descending aorta, these carry, coming off the descending aorta, we have our umbilical arteries. These carry deoxygenated blood back to mom to drop off waste and carbon dioxide and then makes the circuit again. So those are structures of the fetal circulation. It's a nice diagram in your book, but also, as I said, Professor Small should have done this in lab. Let's see. I said, remember the general structures of fetal circulation. So once again, it's going to be the ductus venosus, the ductus arteriosus, the foramen ovale, the umbilical vein, and the umbilical arteries. If you can remember all those, you're going to be fine. Let's see. Roles of gonadal hormones. So in males, testosterone gives male secondary sexual characteristics and is necessary for sperm production. In the females, remember we have estrogen and progesterone. Estrogen and progesterone regulate the uterine cycle. Female secondary sexual characteristics maintain the pregnancy in the uterus, maintain the uterus during pregnancy, things like that. All right, I'm running out of steam, guy, but we're almost there. Stages of development. Okay, so important stuff. Okay, so we have our pre-embryonic stage. Our pre-embryonic stage lasts from fertilization through the end of week 2 or until implantation, whichever comes first. The pre-embryonic stage has 3 specific structures. We have our zygote, which is our fertilized egg, which is our first single-celled form. That is followed by our morula, which is our solid sphere of cells, starting at essentially the 16 cell stage. And that turns into our blastocyst, which is our hollow sphere of cells, which consists of our trophoblast, which is the outer layer, and then an embryoblast, which is kind of our inner cell mass that goes on to form the embryo. It's the blastocyst that implants. Once it implants, it is now an embryo. Embryo lasts from essentially week 3 to week 8 of the pregnancy. It's at this point that the embryo undergoes gastrulation, essentially going from a 2-dimensional disc to a 3-dimensional embryo by doing some folding. And then after starting with week 9, we enter the fetal period that lasts from week 9 until you kick the baby to the curb during childbirth. So those are our main stages of development. Hormones, once again, estrogen, progesterone, testosterone. Pregnancy hormones, oxytocin, which stimulates strong uterine contractions, prolactin, which promotes lactation. Don't forget, oxytocin also does the milk letdown reflex, things like that. Birthing process, remember the 3 stages of birth. We have our dilation phase, which is the longest stage. Can last up to 24 hours in women who've never had children before. Then comes our expulsion phase, which we're actually actively trying to get the baby out. We have strong uterine contractions trying to push the baby through the birth canal. And then we have the placental phase where we deliver the afterbirth, which is the placenta. Make sure you always check the placenta to make sure it's fully intact. If anything gets left behind, it can lead to severe postpartum bleeding and sepsis and death. Let's see. Excuse me, guys. Let me fill in. Take of time for my cold medicine to run out. All right, so uterine cycle. So remember, the uterine cycle is different. Uterine cycle is different. From our follicular cycle. They're two different things. So remember, our 3 phases of our uterine cycle. So we have our menstrual phase, where the functional layer of the endometrium is shed. Then we have our proliferative phase, where the functional layer builds back up again due to a rise in estrogen coming from our ovaries. And we have our secretory phase, where when the follicle forms the corpus luteum, it secretes estrogen and progesterone. That estrogen progesterone builds up the functional layer of the endometrium even more and maintains it in a receptive state for implantation. If there's implantation, great. If there's not, we head right back into the menstrual phase. Oh, other hormones. I forgot a hormone. Inhibin. Right, inhibin is there to reduce FSH and LH production. Remember, it's secreted by the sustentacular cells in the male. Let's see. I think something I left. So we did placental formation, fetal support system. Okay, so blastocyst implants. Remember, it's the trophoblast, the outer layer that goes on to form the chorion, which forms the placenta. Or we have other kind of membranes. We have our amnion that forms. Remember the functions of the amnion. So remember, the amnion is there to provide a neutrally buoyant environment for the fetus, maintain body temperature, kind of act as a shock absorber for protection, but also allows the fetus freedom of movement so its limbs don't fuse together. What else we got? The yolk sac, remember, doesn't actually have yolk, but it does form the first fetal blood cells and also blood vessels. I said the chorion forms the placenta. Remember, the chorionic villi form, which is the exchange surface between mom's blood and the baby's blood. Anything else? Lactation. Remember, prolactin is a hormone that promotes lactation, the actual production of milk. Excuse me. Oxytocin does the milk letdown reflex. It also promotes uterine contractions for labor. That's why, like, oxytocin is actually Pitocin, which they use to induce labor. Let's see. Ejaculation. Okay, so remember, ejaculation, the sperm and its other components are traveling through a series of tubes. So that starts out in the epididymis during ejaculation. From there, it goes into the ductus or vas deferens. from the vas deferens, it goes through the prostate and then through to the urethra. So that's the series of tubes that the sperm travels through in order as it heads out toward the female reproductive tract. Meiosis. All right, so meiosis is how we make gametes. So remember, meiosis is different in males than in females. So remember, in females, we have oogenesis. Remember, in oogenesis, we end up with one ovum if it completes that has 23 chromosomes. In spermatogenesis, which is meiosis in the males, we end up from one spermatogonia with 4 spermatozoa, which each have 23 chromosomes. So that's one of the big differences. We end up with one ovum in oogenesis, but you end up with 4 sperm with spermatogenesis. That's kind of a big difference there. Let's see, anything else? Remember that a spike in LH triggers ovulation. Remember the sperm structure, it has the head, which has the acrosome, which has the digestive enzymes, as well as the nucleus. The mid piece has the mitochondria. The tail has the flagella. Talked about the amniotic fluid. Remember the benefits of breastfeeding. Remember what causes the endometrium to build up each month is the estrogen, stuff like that. So yeah, I am gonna call it a night there, gang. If you have any questions, make sure you email me. But if not, I will see you all. I'll see you all soon. So have a good night. Stop recording.